In intestinal nematodes, Strongyloides stercolis is very important pathogen that route of penetrations are skin and in the form of filariform larva, and sexually transmitted artery infection is also reported. And the diagnosis is rabidiform larva, which is 200 to 250 micrometer long and 16 micrometer in diameter, short buccal cavity, large pulp in esophagus, and prominent genitalium. Uh, this is actually the thread worm and larval migration through the lungs and duodenal aspirate. And entero test may be helpful, which is usually done for the giardia. And infectious to the laboratorians, it is very important factor in this Strongyloides stercolis, and it must be differentiated from the hookworm larva. If you see the picture in the short buccal cavity and the hourglass shape esophagus and genitalium, it's very prominent, and NS also. Strongyloides stercolis is intestinal helminth, and worms are free living. Roundworm, commonly named as threadworm. Infection in man takes place by skin penetration by filariform larva, hatch to the intestine and reach to the anus and cause the allergic pruritus and larva migrans, pneumonitis, and diarrhea. Diagnosis is made by rabidiform larva in a free passing fecal specimen, and the confirmation diagnosis is made by the examining the filariform larva after the culture. Strongyloides stercolis rabidiform larva is both ends are at right angle, and in the stericom filariform larva the ends are at left, only one partial end. The tail portion of rabidiform larva is right and filariform larva is left. Ascaris lumbricard, it is ingested by the infection is by infective OEM ingestion, and mostly from the contaminated water and food. Fertilized worm, which is 50 to 75 micrometer and 40 to 60 micrometer, invade yellow to brown granular yolk, clear zone at either end, may have mammaliated covering and be decolorated. The most common helminthing worldwide, oftenly seen in the with the trichoria, and the eggs required several weeks in the soil to become infective. Larval migration through the lungs. Adult resembles the earthworm, may pass from anus, mouth, or nose. Eosinophilia is you can say the prognostic marker in the identification of Ascaris. Undeveloped unicellular embryo inside, coarse, mammaliated, albuminous covering, thick yellow brown chitin shell. This is one another shape of Ascaris, non-fertilized worm, large, more elongated, thinner shell, no concentric clear zone, can be mammaliated or decorticated, heavy albuminous coating, thin shell, amorphous mass or protoplasm. As we explained, the Ascaris lumbricard, it's a creamy white color adult worm. Female is bigger than male, and contaminated of food and water is the route of transmission. Larvae hatch into the intestine, penetrate the intestinal wall and travel to the liver and heart, lungs and trachea, larynx, and finally reach again into the small intestine and develop into the adult worm. Ascaris pneumonia, appendicite, malnutrition, fever, diarrhea, Luffer syndrome. Occasionally larvae also can be detected in the sputum. Tinea scigenata. The calyx is a tetragonal, 4-cup shaped sucker, no hooklet, and the gravid is longer than 15 to 30 uterine branch per side, irregular alternating genital pore. ooyum, which is mostly seen in the stool, that is spherical and 35 to 45 micrometer, yellow and brown, thick, radially striated shell, and the intermediate host is cow, which is very important, and this is intestinal cystode. And this is the first tapeworm, which is also called the beef tapeworm, and the ingestion of the larva in undercooked beef. Ova are infective for the cattle, but not in humans. So the disease is caused by the larva, which is found in undercooked beef, which is the intermediate host cow. So that's why it is called beef tapeworm. If you see the picture, this is the picture. In the picture you will clearly see the hexacanth embryo inside and radial striations on yellow brown embryo 4 and 3 pairs of hooklet. Tinea solenum is also a tapeworm, but it is a poke tapeworm, and similar like Segeneta, the ova is not infective to the human. No, the Tinea solenum ova is infective to humans, and it causes cysticercosis. And intermediate host is pig, and ingestion of the larva in the undercooked pork meat, which is infective to the humans. But this Tinea solenum ovas are infective to humans. In case of Segeneta, it is not infective. Solenum is also 4 cup shaped sucker and 2 rows of hooklet around the rounded rostellum, and longer. The gravids are longer, and the same as Segeneta ovum. And another organism, and the diagnosis is made by the detection of ova in the feces. Another tapeworm is Dipilobithrum lectum, which is fish tapeworm, mostly found in the fish, undercooked fish meat. And same like the transmission route of all the tapeworms which we discussed previously are the same. The larva in undercooked fresh water fish. It's basically it's badam shape, you can say, almond shape, and no hooklet in Dipilobithrum lectum. This Dipilobithrum lectum cause vitamin B12 deficiency, which is very rare and unique property of this organism, because most of the worms cause the iron deficiency, and this organism cause the B12 deficiency. Another intestinal cystode which is very important, that is Haemophilus nana. It's a diamond shape, 4 sucker, single row hooklet, and round short rostellum. The organism does not have any intermediate host, which is a unique property of this H. nana, and it is very smallest worm in smallest pathogenic worm which is so far identified. And the transmission is of egg in the feces of infected mice and rat. So it's the diagnostic is same like all the tapeworm ova in the feces, and this is dwarf tapeworm. Its adult worm is 2.5 to 4 centimeter long, most common tapeworm, only tapeworm without intermediate host, which we explained earlier. Mainly in the children, they cause the growth retardation. And if we see the picture of this H. nana, it's quite similar to the tinea, but the tinea is dark brown, and it is light, and if you see the colorless shell and filament emerging from the polar thickening, and hooklet 3 pairs, and hexacanth embryo inside. If we here explain that it's the tapeworm Tinea species, which is ribbon-like body worm, Tinea solenum and Segeneta, which is one is the Segeneta is beef tapeworm, and solenum is pork tapeworm. Hermaphrodites parasite are very long enough to be measured in the meters. Infection began with the ingestion of larva. In Cygenata, it's a beef meat, and in the Sulinium, which is from the pork. Both parasites are found in the intestine of infected male, and diagnosis made by the eggs found in feces. Ingestion of Sulinium egg by the male result in the disease of cysticercosis, neuroocular and cutaneous, and disseminated cysticercosis confirmation by the histopathological examination of the tissue by eating history of the pork. And you see the worm pictures here. H. nana, which is Haemophilus nana, and also called Vampirolipsis nana, and Hemonolipsis ferterna, and Tinea nana, and it's also the tapeworm, and it's the smallest helminths cystode, more than 40 millimeter in the length, and do not require the intermediate host, and have 200 segment on the body. Contaminated food and water, auto-infections is there, and in auto-infection, oncospheres hatch while the eggs been inside and penetrate to the villi and develop into stereochode and come out into the lumen and grow into the adult worm. And therefore, the H. nana is the only tapeworm that completed its life cycle without coming out from the definitive host, which is human. Mainly cause the bowel irritation. Diagnosis is made by the detection of eggs in the fecal specimen. Hemolypsis diamonota is primarily a tapeworm of the rat and the mouse, but often infect the humans. It is bigger than H. nana, and rostrum is not armed with the hooklet. Diphyllobithrum lictum is also a platyhelminthes, and it's also the fish tapeworm. It's 10 meter in the length. The body is divided into the head and the which is finger shape, and neck and segment are wider. Eggs are oval in the shape. The head have two longitudinal sucking grooves, dorsal and ventral, which is called bothria. And the parasite completed its life cycle in 3 different hosts. The first Coricidium released from the egg in infective to cyclops, in which it developed into the larva, and the larva containing the cyclops is eaten by the fish, and which is undergo further developmental changes and become infective to the definitive host, which is human. And this parasite have not host specificity. It also infect the various fish eating animals, including the man, causing the abdominal discomfort and megaloblastic anemia, which is B12 deficiency. And diagnosis is made by detecting the characteristic egg and the excreta segment in morphologically. And if you see the eggs, these are shaped like that. Further, Diphyllobium caninum is also a helminths cystode, and this is intermediate host is fleas. It is also called fleas tapeworm, or double pore tapeworm, and cucumber tapeworm. If you see the shape of the organism, you will find that it is a common tapeworm of the cat and the dog. Infection mainly in the children. Diagnosis is made by the egg in the characteristic segment in the feces. It is like that. If you see cucumber shape, and the organism is Diphyllobium caninum. Intestinal trematodes are a very important group in pathogenic parasitology. The first one is Parasitolopsis busci. This is the parasite of duodenum, which is transmitted by the ingestion of Metaserratia in aquatic plants. If you see the location of the adult worm is in duodenum, and it's largest trematode, Fasciola busci. And you see the another one is Fasciola hepatica, which is ingestion of the Metaserratia again on aquatic plant, but the location of the adult worm is bile duct, and similar to the F. busci, and it's also called the sheep liver fluke. And chlorrhoesis sensis is ingestion of Metaserratia in the raw undercooked dried salt or pickled fish. It's a bile duct and gallbladder and pancreatic duct is the location of the adult worm, and the ova in the feces are 29 to 16, and bubbles in upper collar, shoulder, small coma shape, uh, protuberance at posterior and thick small with the tiny spine ciliated mericidium inside. It's a Chinese liver fluke, one of the most frequent infections seen in the Asia refugees. Egg is one of the smallest passed by the human, which is you can see here. The eggs are operculum, which is on the head, and you see the small knob, and developmental stage of mericidium, and thick rim around the operculum called the shoulder. You can find here. Fiscula species leaf shape, it's chocolate brown color, and its life cycle in two different organisms. One is definitive host, that is sheep, and intermediate host are snail. In the first, the adult worm lie into the bile duct and shedding into the feces water. A mericidium hatched from the egg enter to the snail host, undergo further developmental. Snail is the intermediate host, and the Serratia cup-shaped tail come out from the snail host attached to the any object, aquatic vegetation, converted into Metaserratia, mammalian grazing or aquatic vegetation, and that is the cause of infection. And the cum and the ventral sucker are the important characteristic feature of the for the identification, the cone and the ventral sucker. And the man Metaferritia is ingested, and the larva insist into the intestine, proceed to the liver by the penetrating the infected wall and then the liver capsule, and it causes the liver symptoms with fever, and also called phascolepsis. And the diagnosis is characteristic egg in the fecal specimen, and serological methods are also useful. You can find out it's like that. Sometimes it is cut, sometimes it's like open, sometimes it's leaf like.